All right, so right now we will proceed with the in-class activity, and my goal here is to kind of walk you through, how I would actually write the approach section, or start writing the approach section based on the instructions that Mary Kay has just provided to you. So here's the slide from the presentation, which I strongly suggest you download, and then use it as a template to develop your own approach section. So just as Mary Kay, I use very structured approach for a number of different reasons. First of all, it keeps me sane because I can actually navigate through the sections of my proposals. And I also don't see how the reviewer would feel comfortable going through the sections of my proposal, simply because very often we refer to different sections of the proposal. And again, structure is good. It's easy to understand. It's easy to go back and forth. So what I'm doing here, I'm copying this section and moving it to the specific aims file, which I've copied, but not for the purpose of just copying everything. My goal here is to reuse some of the sections related to the specific aims, because we will be restating these specific aims in this document. So what I've done here, as I mentioned, I just pasted these components, and here I will introduce a little bit more structure. What I will do, I will actually gonna copy and paste with something which I prepared before this lecture. And now what you see, what I'm doing is I'm actually changing the font size. I'm sorry, changing the font type. Here I'm making this section bold. I'm emphasizing that this is approach. This way, when the reviewer will navigate through your proposal, he or she will be able to quickly figure out where your approach starts, what is your specific aim one, and what I'm doing here, I'm essentially restating what my specific aim is. In the approach section, I strongly suggest that you take word for word the 2 or 3 line tag of text which you've written in the specific aims. Why? Because this part of the text has to be consistent. You cannot be stating something in the specific aims as your specific aims and then changing it on the fly in the approach section. So all of that has to be exact. So what I'm doing next, because I don't want to be bolding in every part of the text, is I'm again here using Mary Kay's template. I'm underlining the text Rationale, which I will be now expanding on. And again, make sure that you do the same thing for both of these aims. So here I still have copy and pasted text for my specific aim 2, which I'm going to get to in a little bit later. So what I do next, I obviously start with briefly typing the rationale here. And usually what I tend to do is I really try to look from like the broader picture, like what are my 2 specific aims, or what are the 2 rationales for this specific aim. So I try to think through the rationales first for the aims before I start outlining the rest of the stuff. Because once you kind of know what you're trying to do, the rest will follow. So again, just to remind you all, what I've done here, I actually modified this figure from what we've drafted on our previous lecture. And here we're drafting this proposal based on the work of Wastelater and co-workers for a really cool magic, I would say. Why? Because in his papers, they don't explain, or they don't really explain the nature of why this magic effect happens. So let me walk you through this effect. So according to their works, and they've done more than just that paper that we reviewed in class, which is shown here, what happens is if you have these nanoparticles which have core structures of a few nanometers, so these are very, very small particles. And in case if you have very large decorative shells, something on the order of tens of nanometers, and if you tag them in a way that they start forming assembled structures, self-assembled structures in case of viruses, they haven't done the coronavirus, which we are proposing here, but they've done other viruses. Magic happens. Suddenly the relaxation properties of the bulk solvent change. So to me, this the reason why I'm calling it magic, because the number of functionalized nanoparticles haven't changed in the sample volume, yet the interaction between the nanoparticles have changed because now they became very close to each other. So this concept in one of the seminal papers, they called magnetic switch. So I will be emphasizing this concept in my rationale for how this method works. So what I'm drafting here, I'm drafting the following text. So for our aim 1 rationale, for our specific aim 1, which is the development of functionalized nanoparticles and quantification of relaxation properties of the paramagnetic biological materials, my rationale is going to go as follows. We propose to employ, and by the way, what I'm doing here, I'm not copying and pasting the text for this from the specific aim, because what we had in the specific aims was very, very condensed. Here we definitely need to expand and explain the rationale behind this specific aim. So what I'm drafting here, and again, this may not be perfect, as Mary Kay said, the key point here is to put your thoughts on paper. It's significantly easier to improve on your writing rather than put all of your thoughts on paper. Don't be afraid of having too much writing. It's a lot easier to trim down instead of expand bits here and there. So my text goes here as follows. We propose to employ previously developed chemistry of magnetic nanoparticle functionalization to create new nanomaterials, because they will be sensing COVID-19, that could selectively bind COVID-19 virions in biological media. This is a typo. The binding selectivity is achieved by the antibody motif, which is conjugated to paramagnetic nanoparticles. So here's my figure, to which I'm going to be referring to a little bit later. And again, if I want to go deeper, I could provide additional details here and add arrows to be able to say that, you know, this is my antibody and so on and so forth. Nevertheless, we continue. The binding selectivity is achieved by antibody motif, which is conjugated to paramagnetic nanoparticle. Once this nanoassembly, and because this particle is nanoassembly, binds to the virion, or virions, I should rather say, it causes dramatic microscopic change in T2, relaxation of water molecules of bulk media, which can be detected using NMR spectroscopy or MRI imaging techniques. And then I say the overall concept of our approach is summarized in figure 4. Why this is figure 4, why this is not figure 1? Because I had my background section. So I'm kind of expecting that I had 2 or perhaps 3 figures in my background section, and this is already section 3. So naturally, this will not be a figure one. One other thing which I also would like to emphasize based on what I've seen in the homeworks which were submitted today is really how do you embed the figure in Microsoft Word. So in Microsoft Word, my suggestion is to insert the text box. That's what I do, and there are many different approaches, but the reason why I insert the text box is because, and let me show you an example of introducing the text box. Okay, is that really not happening? Oh my God, this is really slow. Okay, so this is how we introduce the text box, and then we insert the figure inside of this text box. Why do I use this approach? Is because, well, most of my figures, I'm trying to condense the information, and I'm really trying to preserve as much space as possible for the text and really trying to maximize the use of the space. So most of my figures, they will not be full page width, and inserting figures inside of these text boxes really helps to achieve this goal. Also, as I'm going to be writing and rewriting this text, these boxes, they will move over along with my text by keeping the relative positions. What I also do inside of these text boxes, I type in the figure caption within that same box. And again, there are different ways of doing this, but I find this way helpful because the figure caption sticks around with the figure. I've seen quite a few actual proposals during the review when the figure caption has migrated over, and this makes the proposal reading very difficult. You have to specify the figure number. You have to say figure 4, figure 25, whatever. If you're not given this, and it will be very difficult for you to refer in the text to what you're referring. And I've seen quite a few of these errors already made during the submission. You're not really cross-linking this information. And what I do additionally, as you see here, I'm making my figure statement in the text bold. This way, when somebody reads the figure and realizes this is figure 4, they can go back to the text to see where this figure is being described. And vice versa, in case if someone is reading the text and, oh, hearing this figure 4 statement, he or she can quickly go to the figure, identify the figure, because this text is boldened, it's much easier to see, to be able to find out which figure we're referring to. One other comment here is, as you're going to be writing these sections, when you're describing the figure, and in this particular case, the figure is very conceptual, make sure to have the figure at the part of the text where you're discussing this figure. Why? Because it's very difficult for the reviewer to read your text when the figure is, for example, 2 or 3 pages away. This way, the reader will have to flip over. When this happens, we as humans, we lose our attention, and because our memory span is relatively short. And as a result of that, it makes reading the proposal more and more challenging. So obviously, we're not going to have preliminary results in this proposal because we haven't done anything. And I'm going to actually not necessarily delete this section. I will leave here alone because this is kind of like background. But I will still add a few sentences here along with my rationale, explaining what this method is based on and where do we want to take this method with respect to the experiments that we will be planning on doing. So additionally, what I would like to mention here, as you're going to be writing your approach section, you will be using referencing software, you will be inserting references, you will be inserting perhaps additional references. This is how the text is going to expand. So part of the reason why Mary Kay and I gave you this additional time extension through Tuesday night is really with the realization that this is a significant amount of the additional writing. So please use this time wisely. Please plan your activities to be able to distribute your workflow. Don't plan on crunching through this assignment through one evening. It's fundamentally not possible. You cannot do this in 4 hours. Just bear that in mind, or even 5 hours. This is hard. So what I'm writing here is Wayslater and co-workers have demonstrated the feasibility of viral-induced self-assembly of magnetic nanoparticles for detection of viral particles in biological media. Here I'm inserting this citation, which I will then format properly. So what I'm doing here, I'm inserting the text from my endnote. Overall, the approach is based on 2 key principles. And what I'm going to do here, I'm going to cite another paper, because the paper which we reviewed was not exactly their seminal paper. This was a follow-up paper based on their Nature Biotechnology paper. So here I'm going to copy this paper, move it over to my Word document, and I'm going to insert this citation for their overall approach. So, as we move along, I keep on adding our citations. First, tagging of paramagnetic nanoparticles with virus-specific surface antibodies allows for the functionalized nanoparticles to selectively bind to virions. Second, paramagnetic nanoparticles are small. And here I'm providing some additional specifications. Actually, they're 3 nanometers paramagnetic core with decoration spanning to 45 nanometer diameter. The reason why I'm specifying all of this information is because we're in the approach section. This is where the rubber hits the road. This is where we're doing all of these nitty-gritty details. Second, the paramagnetic particles are small and have relatively small effect on overall T2 relaxation of water. Once these functionalized nanoparticles self-assemble with virions. The self-assemble with virions. The resulting larger, these are still sub-micrometer structures, the resulting significantly alter T2 relaxation of water, of surrounding water molecules. So again, because I'm kind of describing background, for every sentence which I'm going to be typing in here, which is not my idea, and for which I feel I should back up my claims, I will be citing literature. Why do we do this? We don't want to end up with the reviewer criticizing us for something which hasn't been done or hasn't been presented. It's okay to recite things even if you're repeating the same reference over and over again. You really need to give the reviewer where this idea is coming from to back your claims. So that's kind of the overall idea for the scientific rationale. And now we will switch over to a more detailed outline for this specific aim. So now we're going to go through the proposed experiments. And again, our idea was to be able to develop these nanoparticles for sensing of coronavirus particles. So proposed experiment number one, what do I think I will be doing? Well, most likely proposed experiment number one will be synthesis of functionalized nanoparticles with functionalization by COVID-19 specific antibody. And as you see, as I'm drafting, my text is not perfect. You know, here I'm saying functionalization. I'm going to change this word by decoration. And again, you're going to go through the same issues over and over again. It's okay, because you can either fix it quickly or fix it later. So that will be my experiment number one that I'm thinking about. So once I'm going to make these materials, what I'm going to do next? So most likely what I will have to do, I will have to somehow characterize these materials. And here I'm actually going to use exactly the same method as Wastelader and workers use. And again, you're welcome to come up with your own methods from some other papers, but here I'm actually going to use the exact same method, which was, I believe, AFM, which stands for, I'm forgetting what it stands for. Just give me a second, I will switch over. Oh,
are you talking about atomic force microscopy?
Yes, atomic force microscopy. Thank you very much, Mariki. So proposed experiment number two, characterization of the synthesized materials by atomic force microscopy. Why? To confirm their size distribution. And here it's a good idea to actually state expected outcome. So again, I will be expanding on the details of the experimental section, but my expected outcome here will be monodispersed distribution of nanoparticles of approximately 50 nanometers. Why? Because this is my goal, to create nanoparticles which are similar in size to the virions of coronavirus. This method, one of the reasons why this method is so cool and it works so well is because the particles that we're trying to conjugate, they're similar in size to the viral nanoparticles. And as a result of that, it is possible for them to form this interesting nanostructures, which effectively change the magnetic relaxation properties of water and produce magic. In case of these nanoparticles would likely be significantly smaller and significantly larger, it wouldn't work so beautifully. So this is my expected outcome. I'm gonna underline that to emphasize that this is different from what I'm trying to do here. So I'm also gonna underline this text as I'm gonna be typing everything else again. So proposed experiment number 3. So once I characterize my nanoparticles, I will be performing the relaxation properties of these nanoparticles. Relaxation quantification of nanoparticle materials. What else can I do to make this approach more robust and reproducible? Well, maybe what else I should also do, maybe I should also check the stability of particles, right? If my particles are not going to be stable, in case if they're going to be degrading too quickly, then I'm going to be perfect platform for the experiments that I'm planning to do later. So I'm actually going to introduce my 4th experiment here. So this will be stability validation of the developed materials. Again, based on what I read in the literature, in some of these papers, these materials should be stable for weeks to months. So again, here I'm going to specify my expected outcome. And my expected outcome, material stability on the time scale of many weeks. The reason why I'm not putting many months is because I may not have sufficient amount of time during the time scale of this project to be able to perform testing of these particles for many, many months. So I like that. So once we perform the relaxation quantification of the nanoparticle stability validation, we should also think about the potential pitfalls and alternatives. Well, one of the potential things that can go wrong, maybe our chemistry is not going to give us a uniform distribution of the particles. So why is that important? Well, it's really important to how the properties are going to be changing and so on and so forth. So, you know, I'm not going to be drafting like excessive amount of details here, but you know, the things that I'm going to be kind of thinking about in terms of the outline is size distribution. So really, the way I'm kind of thinking about this section is what can go wrong. Size distribution. We don't know if our functionalization here is going to be specific or non-specific, so I'm going to leave it alone. My next specific aim, which I'm actually going to be copying from the things down below, is the testing of the developed materials. So I'm going to copy this text, move it over to specific aim number 2 here. And what I tend to do as I write this outline, I start to underline things, I start to format them properly. Why? Because I want to make sure that my formatting later on is going to be consistent. So this is not finished outline, but it gives you a point, a perspective to keep the structure. And the reason why I tend to do it during the outlining section is because everything is very close to each other. It's very easy to fix it instead of going from page to page, page to page later. So this is my specific aim number 2. Where I will be performing testing of the development nanomaterials in vitro. So this is really a validation of the development nanomaterials, validation.
So Ed, we're at 5:37, so we should probably finish up pretty soon.
Fantastic. So this is good. Let's kind of leave it here. So
Okay.
again, well, my suggestion here is to maintain the structure, and it's all going to be good.
Great. So we had a question. So any questions about the approach? Because there's one question about the aims page that I wanted to bring up, that one of the students, Fuad, asked. And he asked, should the aims page include your approach? So I don't know, Ed, if you want to answer that or if I should answer that. But yeah, there should probably be a little bit of information about the approach, but the aims page is a page. So you got to keep it kind of short. I think our advice was to add just enough detail so that the reader understands that there is a viable approach that you're thinking about, but not enough detail to make it so long that it would take up too much space, right?
Right, and my take on this is make sure that you do your mandatory things on the specific aims, discussing the significance, discussing the broader perspective, defining these specific aims. If you've got space left, please use it for something else. Don't leave your specific aims page three quarters long. This is your time to sell your work. If you're not doing a good job on that, or if you're not taking advantage of this space, you're not helping yourself. Obviously, don't try to overload that page. That's another bad way of doing things, trying to super condense everything, because then it's like, you know, solid blocks of text. But just, I mean, my takeaway is like, if you can drop a few sentences regarding your approach, please do, especially if the approach that you're going to be taking is like very novel or very cool, or has some degree of novelty. If this is something that helps you to achieve your goal, to convince the reader to fund this proposal, please do that.
Okay, and there's another question. What if I have more than one characterization method? Should I put them all under one subsection, or should I separate them, or should they be separated?
It really depends. It really depends on what you're trying to do. My suggestion is, in case if you can maintain the flow of your proposal and break it down into sections, please do. In case if this is very difficult to accomplish because these things have to go in tandem, then don't.
Yeah, so my answer to that would be to think about, if you have more than one characterization method, if they all accomplish the same goal, like you're just having multiple characterizations to kind of show reproducibility, or to show the standard in the field as you characterize the molecule in these three ways. But if those three ways have different goals, then you might choose to put those in different subsections, or you could put it under one kind of goal, which is characterize my materials, and then in three kind of even more subsections, you can put those three characterizations. I've seen it both ways, and I guess this is maybe a style issue. What's your style? What do you like? What would make sense to you if you were reading it? Think about your reader. So it's a great question. I'd say there's no right answer. Just do what you think would be the most clear to the reader.
I like this question for another reason. So the reason why I like this question is, there is frequently more than one method to perform characterization. So as you're going to be writing your text, what you should be asking yourself in the context of what Mary Kay discussed in the context of rigor and reproducibility, what you should be asking yourself is there another robust technology to be able to perform quantification of your method, of your material, or characterization of your material? In case if there is, in case if you can I can do it with FTIR. Maybe I'm not going to get all that fine information, but hey, this is another completely different technology to be able to perform characterization. You're going to be dinged very hard on an IH study section in case if there is a different method, like very robust method, and you decided to skip on it. So, and again, it helps to convince yourself in the paper or in the grant application that whatever you're doing is going to be unbiased, and it's going to deliver you the conclusions that you are hoping to achieve. So great question.
Yeah, one other question. Are we not supposed to write published data in the preliminary data section? What should we do with that section? So as I said earlier, you do not need to have a preliminary data section in your independent proposal because you do not have preliminary data. When we start talking about your prospectus, which is the data, which is the work you're doing in your research lab, then you should have preliminary data. And we can talk about that at the end of the class. But for this independent proposal, you do not have preliminary data. You do not have that section. You will have 3 sections: rationale, proposed experiments, and pitfalls and alternative approaches.
Right, and just to echo that, there is a type of NIH proposals which absolutely prohibits preliminary data.
Yeah, right.
The NIH not usually follows the rules, but on paper it says you cannot have any preliminary data. So this is not hypothetical task that we're giving you. This is very much real. There are these high risk, high reward proposals or funding opportunities which exist where you don't have preliminary data, yet your emphasis here will not be on your preliminary data, but rather on background and literature. So try to back up your vision, your approach really with what's been done in the literature.
Okay, I think we should stop there. We've gone a little longer than we had anticipated. So we need you guys to get to your breakout sessions and really start talking about your